Hello guys, I'll be talking about enzymes 2.5 today. So enzymes are mostly globular proteins, which helps to speed up biochemical reactions by lowering the activation energy. There are three stages involved in the mechanism by which enzymes speed up reactions. So first, the substrates, which are the substances or reactants that the enzyme works with, have to bind to the active site of the enzyme. And the active site is a sequence of amino acids on the surface of the enzyme that accounts for its biochemical function. And for them to bind, they have to make contact with each other. In other words, collision with the right orientation. After the substrates bound to the active site of the enzyme, the second stage begins. The enzymes will catalyze the reaction by lowering the activation energy, and the substance will change into different chemical substances, which are the products of the reaction. Lastly, the products are released from the enzyme, and the enzyme is free to catalyze another reaction. Due to the specific conformation and amino acid sequence of the active site, only substances with the complementary shape and the chemical properties can fit and bind to the active site. And this special property of the enzyme is called enzyme substrate specificity. And because of this, one enzyme can only catalyze one reaction. And since our bodies carry out thousands of biochemical reactions in the cells every day, we also processes a huge number of enzymes. And for some enzymes, the presence of cofactors and coenzymes will assist the process or is essential for the enzyme to function properly. So, enzymes serve as biocatalysts that help to speed up the reaction time of biochemical reactions. They speed up the reactions by lowering the activation energy, so it's easier for the reactants to move on into the transitional state. Enzymes are not consumed in those reactions, even though their conformations might change ever so slightly during the process. They will return to their normal shape after the product is released. And the change in free energy between reactants and the products will not change as well. In case you're wondering how the enzyme lowers the activation energy of reactions, I will talk about two of the many ways that enzymes do this. The first one is that when the substrates bind to the active site, the substrates are brought closer to each other and hold in a way that makes them easier to react in the right orientation. And this is common for enzymes to catalyze synthesis reactions. Another method involves the presence of cofactors, which are metal ions that assist enzymes to catalyze the reaction. They have electronegativity, which attracts the electrons from the bonds between the substrate molecules, which weaken the bond and speed up the reactions. And if you are still confused about this topic, please check out the link below that I post in the basic info. It's a video that focuses on how enzyme affects activation energy. So there are 4 factors that will affect enzyme activities: temperature, pH, substrate, enzyme concentration. Please know how to draw any of these relationship graphs on your IB exam. And there are 2 ways to draw the graph about the enzyme concentration depending on the question. For the one I have in this slide, it's the graph with axis substrates, and I'll talk about the other type in later slide. Temperature. Let's focus on temperature first, because temperature is a measure of the average kinetic energy in a system. When temperature gets lower, the average kinetic energy of the system drops as well. That means the enzyme and the substrate will move slower, so the chances for them to collide with each other decreases, thus lowers the enzyme's activity. And when temperature drops to a certain degree, the enzyme activities can stop altogether. However, denaturation will not happen in low temperature. So once the temperature gets back to the normal range, the enzymes can work again. On the other hand, when temperature gets high within the range of heat tolerance, enzyme activities will increase because higher kinetic energy leads to more chances for substrates and enzymes to collide. However, when the temperature gets too high, it will cause denaturation in the enzyme. Since the bonds in enzymes that fold them in functional structures are relatively weak, high temperatures will cause vibrations of the molecule and break the intermolecular forces that hold the enzymes in its functional conformation. So it will change the geometric shape of the enzyme, including its active site, leaving it no longer functional. That's why you see on the graph the enzyme activities decrease when the temperature are higher than the enzyme's heat tolerance range. So another factor that would influence the enzyme activities is pH. So every enzyme has its own pH tolerance range and its unique optimal pH range. When an enzyme is working at its optimal pH range, the rate of reaction will be at the highest. However, when an enzyme works at a pH environment deviates from its normal range, the enzymes will be denatured by protonation. Beyond a certain point, the enzyme activities will cease altogether because all enzymes are denatured. For substrate concentration, the first graph explains why increasing substrate concentration will increase the rate of reaction. When there are more enzymes than substrates, enzymes are not all used because there is not enough substrates. Also, it's harder for the substrates to collide with the enzyme when there is few substrates. When the substrate concentration increases, more enzymes start to work, and it is easier for substrate to collide with the enzyme. As you can see in high concentration, every enzyme are occupied by a substrate, so the rate of reaction has reached its highest point, and that reaches the point of saturation. Even you increase the substrate concentration at this moment, the rate of enzyme activities will not rise anymore because enzymes are saturated already. Imagine that the difference between 10 workers and 1 worker working at the same project, while the amount of work that is required to finish stays constant. The group that has 10 workers would finish it at a much faster rate than the 1 worker group. And this is an analysis between the enzyme concentration effect on enzyme activities. And the plateau in the graph starts when the substrates are all used up. So the substrate concentration becomes the limiting factor of enzyme activity. And this slide represents the 2 possible ways of correlation depending on the situation. So enzymes are proteins, so they're also subjective to change in both temperature and extreme pH environments. High temperature will cause vibrations within the molecules and break intermolecular bonds that accounts for their secondary, tertiary, quaternary levels of structure. So the shape of the enzyme will be changed, thus it can no longer fit to the substrate. Another factor is extreme pH, since pH is a measure of hydrogen ions concentration. When the hydrogen ion concentration is higher or lower than the level at which the enzyme normally works, the conformation of the enzyme is altered. Beyond a certain pH, the structure of the enzyme is irreversibly altered, so the enzyme loses its biological function. Again, denaturation is a process in which proteins, which in this case enzymes, lose the quaternary structure, tertiary structure, and the secondary structure, which is present in their native state, by application of some external stress or compound such as strong acid or base. And remember, denaturation do not break peptide bonds, so the primary structure will remain intact. Okay, moving on to different ways of inhibition. So competitive inhibitor resembles the shape or mimics the shape of the substrate where it binds to the active site. So you see that the triangle shape here and here are similar. However, they do not have to be similar in their total structure. So they compete with the substrates to enter the active site. And when they bind to the active site, they will not be reacted. This type of inhibition is reversible because competitive inhibitors bind to the enzyme through weak non-covalent interactions such as ionic bonds, hydrophobic interactions, and hydrogen bonds. Because reversible inhibitors do not form any chemical bonds or reactions with the enzyme, they are formed rapidly and can be easily removed. And as substrate concentration gets higher, competitive inhibition can be pushed out, and the enzyme will return to its normal activity. Many drugs are examples of competitive inhibition as they temporarily disrupt with our normal biological reactions. In contrast, non-competitive inhibitors do not compete with the substrate for the active site. They bind to the allosteric site of the enzyme, which change the conformation of the enzyme. Irreversible inhibitors covalently bond to the allosteric site of the enzyme, cause chemical changes to the active sites of the enzyme, and it cannot be reversed. Please remember that competitive inhibition can be overcome by increasing the concentration of substrates, while non-competitive inhibition cannot. Let's look at the graph. So this graph represents the effect on both competitive and a non-competitive inhibition on the rate of enzyme activities. The curve of competitive inhibitor did not grow as fast as a normal enzyme curve initially. However, with the substrate concentration increases, it reaches the same level as the normal enzyme curve. Again, this is because competitive inhibitors can be overcome by high concentration of substrates. However, for the non-competitive inhibitor, the enzymes that were altered by non-competitive inhibitors are permanently inactive. So the overall number of active enzymes decreases. So the maximum rate of a reaction reached by increasing substrate concentration decreases as well. Okay, feedback. Thirdly, there's feedback inhibition. There is the phenomenon where the output of the process is used as an input to control the behavior of the process itself, oftentimes limiting the production of more product. The final reaction in the pathway reacts with an enzyme along the pathway at the enzyme's allosteric site, changing the conformation of the enzyme, thus shut down the pathway and prevent the final product from synthesizing. This type of inhibition is used heavily in metabolism regulation. Feedback inhibition helps maintain homeostasis in cells. So you can see it doesn't have to always go back to enzyme 1. It can go back to any enzymes along the pathway to shut down the pathway reaction. So lock and key model. There are two models that were ever proposed about the basic mechanism by which the substrate binds to the active site of an enzyme. The lock and key theory was first proposed by Emil Fischer, I don't really know how to pronounce this name, in 1894, but it is later abandoned and modified into the Induced-Fit model. The lock and key model suggested that the shapes of the active site and the substrate have to be fixed for them to bind to each other perfectly, just like locks and keys. However, the model cannot be explained with adequate experimental evidence, so the modified theory was proposed. Induced-Fit model was suggested by Daniel Koshland in 1959 in the US. Unlike the rigid enzyme model, the Induced-Fit model suggests that the substrate will alter the shape of the enzyme slightly after their binding, so the enzyme is partially flexible. Only the substrate with the complementary conformation in a sequence that matches with the active site of the enzyme will fit and react. And this model is supported with high resolution X-ray analysis of enzymes and other developed techniques. Immobilized enzymes were first introduced in 1897. They are enzymes that are restricted to a fixed place for use in bioconversion. Scientists found out that enzymes can remain functional even when their host cells are dead. There are many ways to restrict the movement of enzymes, including attaching them to a nonpolar surface, most commonly glass, trapping them in an alginate gel, or aggregating enzymes into a big group. Immobilized enzymes are heavily used in industry right now because they're advantages over dissolved enzymes. They can be easily separated from the reaction, stopping the reaction at the desirable time. Enzymes can be reused because some enzymes are very expensive, so recycling immobilized enzyme will save a lot of money and improve economy. Immobilization increases the stability of the enzyme to changes in temperature and pH. Lastly, It will be easier to regulate enzyme concentration with immobilized enzymes, which in turn can speed up the reaction rate by exposing substrates with higher enzyme concentration. This is required for IB student to know, so I have stated the page number next to the title, and I'll talk about it as well. So lactase is an enzyme that converts lactose to galactose and glucose, and lactose is a disaccharide found in milk. People who don't produce enough lactase in their bodies can't digest milk very well, so are called lactose intolerant. Lactose-free milk can solve this problem. So first, lactase is extracted from a special type of yeast that grows naturally in milk and then purified. They are then incorporated into milk in an inactive form or in pill form. And when those lactase are ingested into your body, they will be active once they are in the right environment, like desirable temperature and pH. There are some advantages. First, lactase helps lactose intolerant people digest milk better. And by breaking lactose down to glucose and galactose, will make ice cream sweeter without adding additional sugar, because glucose is a sweeter form of sugar than lactose. Also, converting lactose to simpler forms will prevent them crystallizing during the production of ice cream. So it gives ice cream a smoother texture. Lastly, bacteria ferment glucose and galactose more quickly than lactose, so it can accelerate the production of yogurt and cottage cheese. Thank you for watching, and good luck on your exam. Please subscribe to my channel if you find it useful, and as I will be posting more biology and chemistry videos. And if you're wondering some of the specific topics, you can check out the links below. Thank you.